proteins, which makes it very interesting to work on, as a design target. And then I'll show you a specific project where we have worked on recapitulating some of the supramolecular features of collagen, this highly organized banded structure that the natural protein adopts, and then a little bit about where we see this project going in terms of designing protein matrices. Okay, so let's start.
Illogical.
Oh, all right, there we go. So proteins. Proteins are essentially these large macromolecular nanomachines composed of anywhere from hundreds to thousands to even tens of thousands of atoms. And they have highly organized structures, and they can do a variety of different functions in biological systems, from functioning as catalysts. So here's an example of an enzyme called amylase. And you can see here there's a substrate bound on the surface. They can function in transport. So a protein that we're all familiar with is hemoglobin, which carries oxygen from our lungs throughout our body, and it carries other gases out and releases them in the lungs. And here you can see in the case of hemoglobin that not only is it this large complex machine, but the machine moves, and the binding and release of oxygen is coupled with large scale motions within this particular protein. There are proteins, for example, that function in signaling that'll take signals from outside of the cell, chemical cues, carry them through the cell membrane, and then start a cascade of processes inside of the cell that would be the cell's response to that particular signal. And then proteins also just function as structure. This is the class of proteins that I'll mostly be talking about today. And proteins can form networks, they can form supramolecular assemblies, and a lot of our tissue strength and properties in our bodies are driven by the organized structures of proteins. So, these are all proteins. All these different molecules that I'm showing you on the screen are proteins. And essentially what unifies all of them is that they're able to sort of function within this particular size regime that is a little bit larger than small molecules, but smaller than larger macromolecular structures that we're able to see, with traditional microscopic methods. And it's this sort of nanoscale regime, which, a lot of you are familiar with, where not only, proteins exist in terms of their size, but it also allows them to operate and do chemistry and undergo physical processes that are not possible at these other scales. And this is really the regime in which life operates. And a lot of the reason why, these proteins sort of function at this size is because of the unique properties at this nanoscale regime. And then also in terms of, our ability as engineers to control the structure of matter at different levels, proteins sort of occupy a unique window. You can make much smaller molecules by organic synthesis. And then as you get to lithographic and printing techniques, you can make things that are larger. But sort of in this nanometer to 10 nanometer regime, it's very difficult to get precise control over structure. And proteins are one way that nature has figured out how to control structure at this particular size scale. And so I'm sure you all know this, but it bears repeating that proteins are polymers. They're heteropolymers of amino acids. They are ubiquitous throughout all organisms in life. These are the machines that drive biological processes. And I say they're heteropolymers because they are composed of over 20 different types of monomer units. These are the 20 amino acids that make up proteins. And what differentiates one protein from another, what differentiates hemoglobin from collagen, is not only the length of this polymer, but also the sequence of the individual amino acids within that. So for example, here, this is a one-letter representation of a protein sequence, where each letter refers to a specific type of amino acid. And then this is the physical representation of that polymer chain, where, for example, this one amino acid would correspond to one letter in this sequence right here. But this is not the actual functional form of the protein. This is sort of the extended polymer that is synthesized inside of the cell. But as soon as this polymer is created, it starts to undergo a conformational search and fold. This is the word that we use in the field of proteins. It folds into a very specific three-dimensional structure. We call this the protein fold. As long as the energetics are favorable, as long as it's energetically, free energy is favorable for folding, you'll make this well-defined three-dimensional structure. And what that does is not only, it not only gives the molecule shape, but it also allows for precise chemical control of specific groups. So for example, the protein I'm showing you here, this is a protein called myoglobin. This is like hemoglobin, but it's found in muscle cells and it binds oxygen. And the oxygen is bound at this particular site right here. There's this chemical group called a porphyrin. And what the protein fold is able to do is to hold one amino acid over here, a histidine, that holds the porphyrin in place. And then the second amino acid over here, another histidine, which doesn't actually bind to the porphyrin, but it's held just an angstrom or so away so that you can get very specific gas binding at this site. And it's only because the protein has this well-defined structure that it's able to make this sort of highly tuned active site for this particular protein. And so this is really where the function comes from. The sequence sort of dictates the order in which things appear in the sequence, but it's the fold which dictates the chemistry and the function. And just for scale here, this is about a nanometer on the edge of this box. One of the other things that makes proteins very attractive, both as for evolution for life, but also as a material for protein engineering, is that proteins are addressable. We can take a specific position in that sequence and we can change its chemical identity. And this is done through now well-established methods in molecular biology, where you take actually the DNA sequence, you start with the DNA and you make a mutation in the DNA. And depending on what that particular region of DNA codes for, that'll correspond to a particular mutation in the amino acid sequence of the protein. So you'll take that DNA, you'll put it into a bacterium. The bacterium will think that it's something that it should make, so it'll start cranking out, synthesizing your protein for you. This is called expression. And then we're able to purify these proteins and study them in the laboratory. And so depending on whether or not the mutation we've made is acceptable within that three-dimensional fold, whether it's compatible with that fold, we may find, for example, that when we change this histidine to an arginine, that there really is no impact on the structure of the protein. There's no impact on its function, no impact on its structure. And these are what we call neutral mutations. And most mutations in proteins tend to be neutral. They don't seem to have any effect on structure. And this allows evolution to sample lots of different sequences for proteins. And that's why proteins have the vast variety of sequences that they do. But some mutations will cause a change in function. That could be a loss of function. Maybe that histidine that's controlling gas binding is replaced with something else that either removes that control or blocks gas binding completely. But the fold of the protein is still maintained. The overall structure is still stable. And so this is the way that proteins can either evolve new functions, or in some cases that can impact fitness and reduce the fitness of a protein. And then some mutations occur in places where the protein cannot accommodate them in the three-dimensional fold. So in that process where the chain is collapsing into a compact state and adopting this highly defined fold, this mutation actually causes disrupts that process. And these proteins will then remain unfolded and will be chewed up inside of the cell and degraded, or sometimes they aggregate. And so this is sort of the process that we as engineers, when we're modeling proteins, we have to think about not only how does this mutation affect the folded state of the protein, but could it lead to aggregation or other problems with the structure. And so when we make these substitutions, when we change the sequence of an existing protein, we're guided by our understanding of biochemistry. So these are the 20 amino acids that make up the alphabet of the polymers for proteins. And so, for example, they can be classified by their physio-chemical properties. So if I were to look at these ones in red here, these are hydrophobic amino acids. They don't like to interact with water, and they're often found on the inside of proteins. And if I were to substitute one hydrophobic amino acid for another, that would generally be considered a conservative or a neutral mutation. Similarly, for ones that are hydrophilic that like to interact with water, and then there are amino acids that are charged. So we can use our understanding of the physical properties of amino acids to guide the kinds of changes. And in some cases, maybe we do want to change the chemical character at a specific position, add a charge somewhere, or remove a charge somewhere. And we can use this chart as a way to kind of think about how we want to modify the sequence. But in some cases, we do this on a much larger scale. We're not just looking at a single mutation to a single protein, but we want to look at thousands of different combinations of mutations and see which combinations of mutations will allow us to evolve a new function or to stabilize a protein or improve its solubility, improve the concentration which you can work with it in solution. And so one of the ways that we start to look at larger sequence spaces and evaluate which sequences are worth exploring and which sequences are probably not going to work is using computational models. And this is something that our laboratory works on and develops. We have our own models, and we use a lot of other tools that have been developed in the field. And they all sort of have a similar architecture where you will take, for example, a three-dimensional fold. This is the myoglobin fold that you want to still work within, but maybe change some of the sequence characteristics of it. And you'll suggest changes to the sequence. And then what the computer will do is look at an atomistic model of that protein with the original amino acid and with the new amino acid. And based on our physical chemical understanding, sort of a classical mechanics understanding of what stabilizes proteins. So for example, harmonic potentials for bond vibrations and for bond angles, non-bonding potentials like Coulomb's law for the interactions between charges. You can go through all the atoms that are within a model protein and calculate the energy of that particular state. And then we can say, does making this change in the amino acid sequence improve the energy, in which case we would expect the protein to stay folded, maybe even have better physical properties? Or have we done something that maybe disrupts the target state that we were trying to make, in which case then our protein will be unfolded? And so we can do this very rapidly on the computer. We can scan through lots of different sequences, and then we can focus in on the ones that we think are worth exploring more. And recently, there's been a lot of innovations in this particular way of working with proteins, and atomistic models and physics-based models for modeling these sorts of changes to sequence are now starting to be replaced by machine learning methods that are trained on large data sets of known protein structures and can make predictions about whether a sequence is going to be folded, what kind of fold sequence will adopt, and so forth. So there's a lot of exciting developments in this area. And then the last thing I want to mention is, one of the things that also makes protein design and engineering proteins easy, or easier, I should say, is that proteins often have modular structure. So you'll have a sequence of amino acids, but local regions of sequence will also correspond to local regions of fold. So this is just sort of a schematic of a protein where there are these three folded domains. And if you have two different versions of this protein, let's say, a protein that comes from, whales and a protein that comes from camels, they have very similar three-dimensional topology to them, but there are enough mutations between them that they have different sequences and different, slightly different functions. What you could then do is, if you wanted to make a new protein that maybe has some features of the whale protein and some features of the camel protein, you can look at the boundaries between these domains, the modules, and you could start to make hybrids, or what we call in the field chimeras of these. And you can build chimeric libraries and screen through them and see which ones are functional, which ones give you the new functions that you're looking for. And this would all be done using the same sort of expression and purification technology that we would use for looking at mutations. So, this is the playground in which protein engineers play, and this is just an example of a project that we did with Jim McCann and Ron Coder's lab at City College, where we designed a novel protein. This protein does not correspond to anything that exists in nature, that also has a novel function. It binds the chemical warfare agent VX. And what was essentially done here is we took this protein fold, the three-dimensional structure made up of 4 alpha helices, and then we made mutations to these amino acids that are in the core of this protein. And the mutations were made such that you built a binding pocket that had just the right shape to bind the VX molecule. But specifically, this pocket was designed not only to bind VX, but to not bind other compounds that had similar shapes, but were different enough that they would clash with this particular pocket. And what you can see here is a device that Ron and his colleagues built that uses this particular protein to sense the presence of VX. And you can see that there's a strong signal response in the presence of Vx. But then for these other chemically similar, but non-toxic compounds, you can see that there's a much smaller response. And so, the ability to control structure at this length scale, allows us to make new machines, to make new proteins that can do these kinds of functions. So let's move away from talking about proteins in general, and let's talk about this one protein, collagen. So collagen is a weird protein. And you can see that right away by looking at its primary sequence, the amino acids that make up this polymer chain. And what I'm showing you here is just 1,000 amino acids of the central region of collagen. And I'm highlighting in here in yellow glycine, which is this amino acid over here. It's a very small amino acid. And then proline in red, which has this cyclic side chain like this. Glycine is generally found in flexible regions of the protein. It's one of the more flexible amino acids. Proline is the opposite. It's one of the more rigid amino acids. And collagen is just essentially a concatenation of glycine and proline. The glycine occurs in this very regular pattern every third amino acid. And then there's a lot of prolines in there. So you can see 33% glycine, 25% proline. And you compare that to a typical protein, which is only about 7% glycine, 5% proline. So something is definitely different about collagen than proteins like hemoglobin and so forth. When we look at its three-dimensional structure, we can definitely see that something is different is happening. So here is the structure of type 1 collagen. This is an animal collagen protein. This is the most abundant form of collagen in our bodies. It makes up about, if you were to take your body and pull out all the protein and dry it down and weigh it, about a third of that protein would be type 1 collagen by weight. So this is the protein that makes up the structure of our skin, that makes up the structure of our bones. And here is one collagen protein. It's about 3,000 amino acids, and you can see it goes from the left of the screen all the way over to the right of the screen. Here's the same mass, but now in hemoglobin. So I'm showing you one hemoglobin here is about 600 amino acids, so I'm showing you 5 of them so that it's a fair comparison. And you can see that there's a very big difference in the compactness of the structure. Collagen is this long fibrous thread, and hemoglobin is this really compact, sort of football-shaped molecule. So because of all those glycines and prolines, something about that very different amino acid content is completely changing the shape of this protein. And if you zoom in on this protein fiber, you'll see that there's actually 3 polypeptide chains coming together. So this is not actually one protein. This is 3 proteins that are each about 1,000 amino acids long, and they're making this helical structure where the 3 chains wrap around each other. And these little red dots over here are water molecules that are essentially sticking to the surface of this helical collagen structure. So, and that's just what's happening at the atomic level, at the level of the single protein. Collagen itself actually functions on multiple scales. So here I'm showing you a drawing from a review about almost 45 years old, where they're looking at the different scales of structure that collagen can adopt. So we were just looking at a single collagen triple helix. That is one of these fibers right here in this central thing right here. And so you get this 3 helix trimer that's then assembles with 4 more to make what's called a microfibril. These are then units within a larger structure called the subfibril. We've now gone from things that are on the angstrom scale, on the 10th nanometer scale, to a few nanometers, and then they make larger fibers. These become parts of organized structures and tissues, and then we have tissues in our body like tendons and so forth that we're familiar with. And so this hierarchic assembly of collagen is essential for its function. And one of the things that makes collagen interesting to me as an engineer is the tools that we have for manipulating proteins are at this level. We can change the amino acid sequence, but the effects of that are going to propagate through all these different levels. And so being able to understand how these perturbations at this level affect structures at this level, I think that's a really interesting fundamental scientific problem.
Most illogical.
All right. So an example of this is the way that diseases of collagen propagate over different length scales. So I'm showing you here, for example, that a collagen triple helix, this is a model sequence where it's all prolines and glycines. One of these glycines here in the middle has been replaced by an alanine. And I'm showing you that here in this sort of orange circle right here. And the effect on the structure is there. So I'm overlaying it with blue, which is the same sequence, except there's a glycine at this position. That would be sort of the wild type normal version of this protein. And you can see that by adding an alanine there is some distortion to the structure. It sort of bends out a little bit and then it starts to come back in. So it makes like a little tiny bubble in the middle of the structure. But then as these collagens start to assemble, if they have this particular mutation, if there is no mutation there, you get these nice sort of packing at the subfibril and the larger fiber levels. But as soon as you have this mutation in there, this mutation which is associated with a disease called OI, osteogenesis imperfecta, you start to see that you get malformed structures at this larger scale. And that continues to progress until you get to the scale of anatomical dysmorphologies. It can cause bending of the bones. It can change the mechanical properties of the bones. During development, bones end up being significantly deformed. So how do mutations at this atomic level affect mechanical properties and structural properties going through all these different length scales? I think that's a fascinating problem and an important one.
Most illogical.
I don't know why it keeps doing that. All right, okay. One regime of structure that we are particularly interested in is this feature of collagen fibers called banding. There's this, if you look at sort of the nanometer to tens of nanometer scale of collagen, you'll see that there's this very organized structure. So here, a single collagen triple helix, a single collagen molecule is going to be, let's see, it's going to be about, let me do the math here. So about 300 nanometers in length. So a single collagen molecule is going to be maybe about five spans right here. But the width of this is now you're talking about something that is as wide as a few hundred to a few thousand collagen molecules. And so this structural feature that you're seeing here is actually being formed by thousands upon thousands of collagen triple helices coming together and assembling in an organized fashion. And these gaps represent areas where there's either higher density of protein or lower density of protein. And you can see here, if you sort of zoom in, the sort of staggered assembly of individual collagen triple helices, and the gaps between them create these spaces where, in this case, for example, dye can come in and give you a darker stain. And then regions that are lighter, they're completely filled with protein, so you don't get as much of the dye coming in and giving you that stain. And so the question is, what is the origin of this banding structure? And is this something that is important for collagen's function? Well, so we know that there are a couple of ways in which this banded structure affects the biological function of collagen. So for example, when type 1 collagen is in bones, these gaps essentially serve as nucleation sites for mineral formation. And as the collagen fibers polymerize, they're able to nucleate formation of the minerals, and that results in very strong, rigid, load-bearing bone structures. Similarly, when in tissues like tendons, when you have collagen fibers that are associated with each other, you get these proteoglycan links between adjacent collagen fibers. And these links are mediated by periodic sites that show up on the surface of the collagen fiber. So we know that these periodic structures that collagen adopts affects the physical properties of bone, affects the flexibility of tissues like tendons. It also affects the ways that cells interact with these materials. If you think about collagen making sort of a basement, a sort of surface for cells to adhere and grow on, the organized structure of collagen is important for the healthy growth of a cell. And so here's an example where they're from a study in 2007, where they took a petri dish and they either allowed collagen to polymerize and form these banded structures, like you can see here in this inset, or if they were rapidly deposited and not allowed to assemble, so you still get a fiber structure, but you don't have that additional sort of banding morphology. And then you put a nascent osteoblast on this. You can see that if it's growing on the banded structure, the cell becomes polarized and it starts to grow in a specific direction. This is a cell that is at home on the substrate, and it's going to stick and it's going to differentiate. Whereas this is the same type of cell, but you can see now this one's kind of bubbling and angry. It's not able to get a good purchase on the surface. And often, cells that undergo these sorts of morphological transitions are in what we call a neoplastic state. They're essentially going to, detach from the matrix surface, and it's possible that they can become sort of pre-oncogenic cells because of that pore interaction. So there's a lot of interest in understanding how do cells recognize these organized structures, and what is the way in which these organized structures control the biology of the cells themselves. So we've been interested in trying to design synthetic collagens where we could model this sort of supramolecular assembly and see if we can understand some of these biological processes. So there's been a lot of work in this area, and one of them has been in the area of sort of peptide self-assembly. Collagen is over 1,000 amino acids in length, so it makes these really long fibers. But one of the easy ways to study collagen in the lab in a very productive way is to just make fragments of collagen, small peptides of about 30 amino acids in length that still have this proline rich and glycine rich sequence. But now you can, for example, play with the sequence a little bit and add a positively charged group on one end of the protein of the peptide and a negatively charged group on the other end of the peptide. So you've essentially made this large macro dipole. And then these, as they assemble in solution, will assemble in a head to tail manner, as was at least what is modeled here, and will give you this sort of banded morphology right here. And now you can see that because this is a small peptide, it's only about 30 amino acids, so it's only about a nanometer or two in length. The dimensions of the banding are fairly small, right? They're only around 18 nanometers. Whereas in type 1 collagen, in the structure we saw before, it's over 67 nanometers. So it's a much larger structure. But you are able to get this sort of this banded structure. And there have been a lot of designs since that use the same sort of charge pattern. And some of them make banded structures and some of them don't. So the rules are not quite so clear for this, but we are able to understand some of the self-assembly process from these systems. And then another very promising system that was developed here at CUNY at Hunter College by one of your own faculty, Yujia Xu, is this collagen protein, this mini collagen protein, where domains from animal collagen are flanked by these proline-rich domains, and they form these sort of staggered assemblies. And you can see here from this electron micrograph that same sort of banding pattern. And so, we were inspired by these particular designs, and we were trying to make something where we could achieve the same sort of banded pattern, but we also wanted to be able to control the binding and function of cells to these banded structures. So we wanted something where we could control the function and the chemistry of the collagen assembly. We also wanted to be able to control the shape and mechanics. Could we control the dimensions of the fiber? Could we control the spacing of the banding? We wanted the assembly to be rapid and robust. So we wanted to be able to do this repeatedly. And we wanted to be able to make large amounts of the material. We wanted to be able to express it easily, be able to purify it easily, so that we could now have sort of a platform for playing around with all these different parameters and making something. So this was really a biotechnological exercise. Could we take this? Could we build a prototype of a banded collagen fiber and then use that as a way to ask questions about collagen biology? And so the strategy looks like this. We made synthetic sequences where we follow the pattern of natural collagen, where you have 2 amino acids that are often proline, X and Y, and then a glycine every third amino acid. If you make a sequence with that repeat, they will assemble into a triple helix. But now we had 2 types of triple helices. So remember I talked about how proteins are modular. So we developed 2 different modules for this collagen. One which we call an adhesive module. These are the ones that stick together. These are responsible for the higher order assembly. And then the other, which is the functional module. This is the one that's going to control, the length of between bands, or the interaction with specific cells. And if we combine these then in a specific way, for example, flanking a functional module with two adhesive modules, then we might expect to see a structure like this, where you can get this sort of like log cabin stacking of collagen fibers relative to each other. And in regions where you would have a lot more, in these adhesive regions where you have a lot more protein per unit length, we would expect to see, these one color band. And then in the regions which are open, where dye perhaps could go in, then we would expect to see, a darker band there. So we would expect to be able to see this structure if the designs were successful easily on a microscope. Okay, so what I'm showing you here is an example of one of these designs, and I'll go through this one in a little bit of detail, and then I will, the rest of them will essentially be variations on this particular design. So one module here, the adhesive module that I'm showing you here in gray, we call P and then followed by a number, and that number represents the number of repeats of a glycine, proline, proline triplet. So a P10 would be 30 amino acids long, okay? So that's the adhesive domain. Then the functional driver domain in this case is actually a fragment of a bacterial collagen. So this is about 81 residues from a naturally occurring collagen in bacteria. The natural function of this collagen is really for bacteria to stick to our collagen. So it secretes these collagens and it uses them to adhere to our extracellular matrix. And we wanted essentially something which wasn't, specifically designed for some of the biological tests that we're going to put it through. And then we constructed a gene which contained two adhesive domains. You can see one over here, and then one at the end, the functional domain in between. But before this, we added what's called a folding domain with the V domain. This is from also the bacterial collagen. And this will bring three polypeptide chains together and nucleate the formation of that triple helix. And then prior to that, also, we have a polyhistidine tag, which we can use to purify this protein. And then between the folding domain and the actual protein that we wanted to work with, we included a protease site. So this is a specific amino acid sequence where we can add another protein that will cut this first part off. We can then separate this from the protein that we want to work with, and then this will self-assemble hopefully into the structures that we wanted to study. So here's an example. We call this P10, B for it's the B fragment of this bacterial collagen, then another P10. And you can see here from this micrograph that it makes these banded structures. And this is, on a 200 nanometer scale. So these fibers are maybe a few hundred nanometers in length, and about, 10 to 15 nanometers in width. And we get this robust banding. And we see this again and again with multiple preparations of this protein. It's relatively easy to purify, and we get repeatable assembly behavior. So now can we start to play around with this domain architecture and tune, let's say, the structures of these? So one thing we could do is we could leave the adhesive domains the same, but play around with this piece in the middle. So for example, what happens if I take, instead of the B fragment of bacterial collagen, the A fragment? So it's a different sequence, but it still has that same collagen glycine XY repeat. We can see that again, we get now with P10A, P10, a banded collagen fiber. If we change the length of the insertion, so instead of inserting one B-type domain, we insert two. So now we have, instead of 80 amino acids, about 160 amino acids in between. We still get a banded fiber, but now what you can see that's happened is the light bands are the same width, but the dark bands are now twice as long. And if we insert three domains, the white bands are the same width, but the dark bands are 3 times as long. And you can see that here in these histograms of sort of the bandwidths taken from multiple micrographs, and the light bands corresponding to the adhesive domains don't change in their width distribution, but the dark bands, which correspond to this insertion that we're putting in, its distribution of lengths scales with the length of the polypeptide that we've inserted in there. So we can control the dimensions of this dark band by changing the length of the functional driver. So there was a question, does the V domain help in nucleation? Will the 3 helix stay helical after the V domain is cut off? The answer is yes. Yeah, so once you're able to bring the 3 chains together, the helical domain does 2 things. One is it sort of nucleates the folding of the triple helix, but it also controls the relative registry of the 3 strands. In the absence of that V domain, you could imagine the 3 strands of the triple helix assembling in different sorts of offsets. You might get more heterogeneity in terms of assembly. So it does a lot to both control the homogeneity of the assembly, but also in nucleating it. But once you cut it off, the fiber stays assembled. So similarly, we can leave the functional domain the same, but now we can change the adhesive domains. So for example, here, instead of using these long proline regions, we now use a charged region, kind of like we saw in that peptide study. We have a basic domain in the beginning, an acidic domain at the end, and now you would expect them to attract each other kind of like magnets and assemble in a staggered fashion. And we can see here we get these banded structures again. Here's with the proline, except instead of 10 repeats, we have 8 repeats, or we have 12 repeats. And if we compare these 3 designs with each other, we see that now the dark band stays about the same, but the light band's length changes depending on how long these adhesive domains are. And when we look at all the different designs that we tried, over 2 or 3 dozen different types of constructs. And if we plot the observed bandwidth, so measured from these micrographs, looking at the images, against a calculated bandwidth, and the way that we calculate what the width of a band should be is we look back at the molecular structure of the triple helix. And we ask that if you were looking along the triple helix axis, what is the rise per residue for one amino acid along this? So if you're going from like amino acid I to I+1, you move forward along the triple helix axis by about 2.8 angstroms per residue. So we can then take like an 81B domain or a 30 amino acid P10 domain and calculate what its length should be. And you can see that they fall along perfectly along a line. And in fact, this dotted line is not a fit. That's essentially just y equals x. So I've set that equal to, just to show you that this is matching perfectly. And here is animal collagen, that type 1 collagen with the 67 nanometer banding. And with an ABC insert, we can get very close to that with these synthetic collagens. So we can access these sort of morphologies that are on the same scale as biological systems. Okay, so I'm going to show you now, the images I've been showing you so far have been electron micrographs, transmission electron micrographs, where you're looking down at the sample and you get a 2-dimensional sort of visualization of this banding pattern. We also did cryo-EM of these, and that allowed us to see whether these bands have a 3rd dimension. Do they have height? And this was done essentially by assembling these fibers and then freezing them in vitreous ice, and then doing a measurement called tomography, where you deposit the fibers on a surface, and then you visualize it at different angles. And then by integrating the images at the different angles, we can get a 3-dimensional reconstruction of the fiber. So I'm going to show you this one for P10, BP10. Or am I? Oh, here we go. Okay. So first, you're going to see the tomogram itself. We're looking here at one of these banded fibers, and you can see that we're essentially imaging it from about minus 60 degrees to 60 degrees. And now from that, we're able to reconstruct this vertical slice through the fibers, and you can see that the banding goes all the way through from the top of the fiber to the bottom. This is essentially equivalent to doing vertical sectioning of these fibers. And then we can use those images to do a volume rendering. And so this is the actual fiber structure here that you're looking at. And you can see the banded fiber throughout. What's also interesting is we see that with what looks like multiple fibers coming together, we get this alignment between the fibers, which suggests that once the fibers form, these surfaces still may be sticky and allows them to sort of orient relative to each other. And this is something you see with natural collagens as well. Let's see. And I'll just show you one more. This is for the one with the larger insert.
Most illogical.
There we go. So this has two B domains inside of it instead of one. And what you can see now is that these bands are further apart from each other, but it still has that same banded structure. And then if we look at the sectional view, we can see that these bands go from the top all the way through to the bottom. You could also see that there's a little bit of a suprahelical twist at this larger structural level, which is again something we see in natural collagens. And then from this sectioning, we're now able to make a volume rendered model of these fibers. So it was exciting to be able to see them that way and actually appreciate that these are not just two-dimensional fibers that are assembling on the surface of the microscope grid, but these are three-dimensional banded structures. So we wanted to just, stress test the model and make sure that, our design scheme was reasonable, that you have to flank a functional domain with these adhesive domains. So we tried things, for example, like just putting it on the N-terminal end at the beginning. That did not form fibers. But at the C-terminal end, it doesn't form fibers. You put it in the middle, it also doesn't form fibers. If you shorten the adhesive domain enough, so in this case, this is half the length. We have a P5 in this case. We get something that's fibrous, but it's not a banded fiber. It doesn't make adhesive fibers. So it looks like, you need to have a certain critical length of this adhesive domain in order to get well-organized fibers. And so we looked at that a little bit more closely. So one of the things that our group does, this has mostly been an empirical study, but we wanted to see if we could understand why is it that some adhesive domains promote organized assembly and others do not. So we did a calculation where we just took the adhesive domains and on the computer modeled rigid body rotations of one adhesive domain relative to another. This is that angle over here. And you can see that at a certain angle, when the two are parallel, as you would expect, you get a very good association. We're calculating the energy here, like I showed you in that one slide, using sort of a classic energetic force field. And here, for example, you're looking at P domains interacting with each other. You can see how they can make these tight interactions. Here's with the charged domains, you have the basic and acidic regions interacting with each other, and they make these networks of salt bridges between them. What we found interesting, though, was we looked at the designs where they did assemble into fibers that were organized and did not. And we saw that there was a threshold in terms of the energy. So for example, for a charged group, if you had a basic group that was of length 1, meaning only one arginine on one side, one glutamate on the other, we didn't get assembly. That had a very weak energy. If we made it 3, it was stronger, but it was not enough to drive assembly. But when you got to 5, then it was able to promote these organized fibers. Same thing with these proline domains. The shorter one, the P5, did not. But then once we went to P8, P10, and P12, we were able to see banding. So this starts to give us an idea that there's sort of a critical threshold of pairwise interaction, at least, between these adhesive domains. And this is now something we can play with in terms of designing the sequence and specificity of these adhesive domains to better control the structure. Oh, okay. All right, so I think we're starting to understand how we can control morphology, within the construct that we created. Another question is, are these banded structures functional? Are they actually able to talk to cells and direct their growth and maybe their differentiation? So I showed you that example of the fibroblast, which is either happy on the organized surface or angry on the disorganized surface. Could we see the same sorts of phenomena with our synthetic protein matrices?
Most illogical.
Okay, so the hypothesis is this, that a large part of the way that a fiber can dictate the adhesion and the internal processes of a cell is through these interactions with cell surface receptors called integrins. Integrins are a class of proteins that they're these communicators that will take a signal from the outside and communicate it to the inside of the cell. And there are a class of integrins that will bind to collagens, particularly to certain sequences, these GFPGR sequences or GFOGR sequences. And when they bind, then that'll signal a set of reactions inside of the cell. But more importantly, when you have multiple integrins that are close to each other, so this is like one part of the cell right here, and you now have multiple integrins proximal to each other, that proximity will create what's called a focal adhesion on the cell, that sort of local concentration. And that is believed to trigger the actual cellular response. That's what tells the cell it's binding to a biologically compatible surface, and it'll start to cause the cell to settle in and differentiate. And you can see here, people have solved the high resolution structure of these integrin head domains bound to a small fragment of collagen. So they make an actual interaction here. And if you have a banded structure, then we would predict that these binding sites would be aligned with each other. So if you look along the length of the fiber, you would see multiple binding sites close to each other. So we introduced this binding site into the middle of that bacterial collagen sequence B. So it doesn't naturally have an integrin binding site into it, but we know that that's the region that's important for binding. So we just added the 6 amino acid sequence in the middle. And what we found was that for those proteins which did not have banded structures, so this would be a control protein, bovine serum albumin, which is not a collagen at all, just that B domain by itself with the G Foger site in it, something that has flanking adhesive regions, but they're not large enough to make a banded structure, that we would get cells binding occasionally, and that they would, what you're looking at here, by the way, the green is a stain called phalloidin, which stains actin. This is the cytoskeletal protein of the cell. This is what gives the cell its structure and shape. And in blue, you're looking at DAPI, which is just showing you the nucleus of the cell. Whereas now when we get to banded collagens like P10B, P10BB, and so forth, and actual natural collagen, we can see now the cells are spreading out more, the actin fibers are more organized, and we can quantify this. And we can see that as we go from non-banded, these first three, to banded, that the cells seem to be a little bit more at home on these banded surfaces. So we can also look, for example, at processes inside of the cell that suggest that these focal adhesions are being created. So when you get the clustering of these integrin proteins, they'll now bind to a protein called talin, that'll then nucleate formation of vinculin. And then vinculin will start to interact with the actin cytoskeleton. Those green fibers that you're seeing here, those are the stains of these actin filaments. And so what we can see is that in the non-banded versions, we don't see very much expression of vinculin, of this gene, or a transcription of this particular gene. But once you start going to the banded collagens and the natural collagen control, you can see that you get much higher levels of expression. Same thing with actin. Actin seems to be upregulated when you have banded collagen substrates for it to bind to. So it seems to be turning on the machinery that's triggered by the formation of these focal adhesions. And then once you start to trigger those processes, then that leads to downstream signaling inside the cell. And in this case, we were working with bone precursor cells called osteoblasts. And we started to see in these cells some transformations within the cellular physiology. They were starting to turn into bone cells. And one of the indications of that is expression of an enzyme called alkaline phosphatase, which essentially results in the nucleation of and mineralization of bone. And we can see the expression of alkaline phosphatase by this stain, this purple stain right here. And again, you can see that in the banded structures, we have higher expression levels of these. Also a transcription factor that's associated with cell differentiation. We can see that actually in one of the collagen, synthetic collagens, a much higher level of transcription of RUNX2, and then also for the other ones as well. So, we have a toy now to play with in the lab. We have this synthetic collagen system that we can change the domain structure, we can change the functional driver domain, We can express it in high levels. We can make lots of it. We can do lots of different experiments with this. We're still a far way off from really sort of recapitulating the natural structure of collagen. You can see that when I put the two side by side, there's a lot of differences between them. And I think one of the interesting questions here is, to what extent are those differences important? And this is what's going to drive a lot of the future science that we're doing. So the last thing I want to talk about, and I'll just have one or two comments about this, is where are we taking these matrices next? And I think the first is pretty straightforward. We're going to use our ability to control morphology to think about questions of how we can control mechanics. Can we change the mechanical properties of these materials? Can we change the functional potential of these? We can also use these as models for disease. If we know that a particular mutation in natural collagen causes dysmorphology, of tissue and also at intermediate levels of structure, can we find locations within our synthetic collagens to introduce those same mutations and see how it affects supramolecular assembly? But one of the things that I'm really interested in is how do large proteins like this evolve? So then the collagen that we have today is over 1,000 amino acids long, and it's got this very complex supramolecular structure. How did something like that come about? And what we know from looking at the amino acid sequence of this protein is that it must have evolved through some sort of a process of gene duplication and fusion. So you have an original collagen sequence that from bioinformatics analysis was about maybe 1/15 the size. So maybe, about 80 to 100 amino acids in length. And that single domain was then duplicated and concatenated, and it formed these larger sequences. But we don't have very many examples of these original proteins anymore. And so the question is, what did those proteins look like? What did they do? Did they also make banded fibers? Did they do something else? And although we don't have many examples of these in invertebrates today, there's a class of animals that likely are probably still harboring some of these more ancient collagens, and these are sponges. And sponges are believed to be the first animals to have collagen. And in fact, this is where collagen was thought to have first evolved, was in the transition from these free-floating single-cell animals called choanoflagellates. This is the work of Nicole King at Berkeley. And these choanoflagellates are believed to be the predecessors to the first multicellular porifera, to sponges. And the way that they started making colonies and multicellular organizations was by expressing collagens on the surfaces of their mineralized spicules. And it essentially started as a sticky glue that brought multiple cells together. And then over time, they evolved into more organized structures. So one of the things that we would like to do is to now use our synthetic collagens to model this evolutionary trajectory from small sort of single collagen domains to larger collagens, and see what types of structural and mechanical properties come from that. Okay, and with that, I'll end. I want to say that I was only one player in a much larger project. With a project of this scope, we had a lot of expertise coming in. This was largely driven by my former postdoc and now a faculty member in China, Fei Xu, who really did a lot of the development and characterization of these. And Jin Yuan, who was a graduate student with Fei and myself. He spent several years in our lab synthesizing and characterizing these proteins. Excuse me. Dr. Sun at Tongji University, who did a lot of the cell differentiation studies with us. And then a group of collaborators at RUTGERS, including Jean Baum, who did some AFM with us, and then Wei Dai, who got those beautiful cryo-EM images of the proteins. So with that, I want to thank all of them, and I thank you for your attention.